only able to test the TET-R generator being transformed once. We're not sure of the reliability of those results. And still, we have not been able to successfully transform the banana odor generator, so we have not been able to construct our Circuit 1 design. In the future, if we are able to successfully build Circuit 1, our first step would be to perform a gel electrophoresis to see exactly what DNA is in the cell, and we would also perform other results, other tests to see our results. Next, I'd like to welcome Mike Sheets, who will be talking about those steps that we would take and overall our hopes for the iGEM Club in the future. Thank you.
Thank you, Maddie. Hi, my name is Michael Sheets, and I'll be talking about our future plans for our iGEM team and for our Eco-Factory project. To start, we definitely have a lot of room to grow, especially in research and development. This year was a big learning process as we came in with no synthetic biology experience, but now we know much more about synthetic biology and about the iGEM program. Most of our labs were also bioavailability applied labs, such as Eau coli and E. chromi, to help us learn the techniques we need to use for our actual labs. For our first circuit, what we need first definitely is a successful transformation. To do this, we would need assistance from outside help who has more experience in microbial techniques, and we would also need more advanced equipment, and to do this, we would consider contacting a local lab facility or university to partner with them. For our theoretical circuit, the first thing we need, again, is a successful proof of concept, which would be our first circuit, so we know our Eco-Factory project has a chance for success. After this, we would need to transform METU Turkey's part that senses carbon monoxide and outputs COOA, and to do this, we would need to have it made, and we would need to find out at what cost and how long it would take to make this. For our second part, we would like to ligate together a promoter that is activated by COOA, and also with MIT's banana odor generating part. For this, we would consider transforming both parts in separately or ligating all the parts together to do one smooth transformation. For standardization, characterization, and computation, we would definitely want to work more on detecting the inputs and outputs for our system. For inputs for our first circuit, we use doxycycline, which we can adjust the volume to change the concentrations of doxycycline we use. For our second circuit, we would need to use carbon monoxide in an indole-free anaerobic chamber, so that we could. But for this, we would need to use something like an electric carbon monoxide detector that shows the percentages of carbon monoxide, so we can see at what levels our machine works best. For output, we would like to use our. We would like to test using a qualitative smell test and a fruit flies like Barbara would suggest for our first circuit. However, for our second circuit, again, it will be in an anaerobic chamber, so that will be more difficult to test. We would like to try to titrate the isoamyl acetate and to better standardize and quantify our results. For next year, we have a lot of big plans for our team, and one of our largest is team member specialization, which would be that this year sort of everyone worked on everything, and next year we would want to have individuals who are more skilled in areas such as IT, public outreach, contacting businesses, and media. We would also consider having an iGEM support group of individuals who would be available when necessary to help us with areas that we struggled with, such as IT and programming. Next year we would also like to do more efficient fundraising. This year we already had multiple fundraisers and have recently sent out letters to businesses asking them to consider becoming corporate sponsors. However, next year we would like to expand our fundraisers and do much more with that. For outreach and human practices, this year we already got an article in a local pamphlet and aired a commercial on our school news. However, we have even bigger plans for future years. Next year we would like to do a DNA day at our high school where we talk to students about synthetic biology using fun games and hands-on non-lab experiments. We would also like to go to our local elementary school to do a day called Science Elementary, where we talk to the smaller students about science and especially biology and maybe even a basic level of synthetic biology. We also plan to present to our school committee in the fall about our project and our team, and would like to contact our local fire department about local carbon monoxide statistics and emergencies. We also plan to next year to put out an app and another video. Next, I will call up Kathleen Barrett, who will talk about our experience this year with iGEM. Thank you.
Hi, I'm Kathleen Barrett, and I'm going to talk about our experience this year at iGEM. This is our first year in iGEM, so we ran into a few problems. In the beginning, we struggled to come up with a feasible idea. We found that we'd come up with something that was really interesting, what we thought was really unique, and after putting a little bit of research into it, found that some other previous iGEM team had already completed the project. So after a lot of effort and work, we decided to combine one of our ideas with the work of a previous iGEM team to come up with a unique system to solve a different problem. Our next issue is that we had little to no previous experience with microbiology, and we thought this was going to be our biggest obstacle to overcome. However, the BioBuilder experiments and the iGEM headquarters and the iGEM website were wonderful resources in learning about microbiology, and we were able to actually rather easily overcome this obstacle. Our next problem was our access to equipment. Because we're from a rather small school, we don't have some of the more expensive equipment needed to complete many of the experiments done in synthetic biology and microbiology. Fortunately, we were able to find modified versions of protocols that allowed us to complete our experiments, and we were able to design our own versions of the equipment. For example, we made our own centrifuge out of a salad spinner. Our final issue was that we had limited funding. We are an unfunded club, so in order to raise funds for our club, we had to do a lot of fundraising on our own. We were very fortunate that we had a lot of individuals and companies that were willing to donate their time and money to our clubs, so we were able to proceed. Overall, we had an amazing experience with iGEM. We learned a lot about the techniques of microbiology and synthetic biology. We got hands-on experience in the lab that we would not have been able to get in a normal classroom experience. We were able to work together as a team and learn how to think for ourselves and to implement and execute our own unique ideas. This was an amazing experience, and we're very thankful. This concludes, we'd also like to thank numerous companies and individuals for their support and their donations of time, money, and effort. We'd like to extend a special thank you to Alyssa Hennings from Ginkgo Bioworks, Natalie Kudel from MIT, and Kitwa and Megan from the iGEM headquarters, whose time and effort they put into helping us was essential to our club. This concludes our presentation, and we'd like to thank you for taking the time to watch it.
Go iGEM!
Okay.
It's awkward because I think the camera's not off yet.